We've discussed mitosis. Now we're going to look at a related process called meiosis. This is cell division in germ cells. What do I mean by germ cells? Well, germ cells are those cells that make new little humans. So most of the cells of your body, unless they're tweaked really hard, can't make new human beings. But your germ cells exist for the purpose of making new little humans. So if you're female, you're producing germ cells that we call oocytes that are going to make an ovum. If you're male, then you have spermatozoa that are also capable of making new little humans when these two germ cells come together. It takes two germ cells, one from each gender, to make a complete individual. These join together in a process called fertilization. And so if a fertilized egg is to have the normal DNA complement, then each one of those germ cells needs half the normal amount of DNA. So the normal DNA content of all the cells of your body that aren't germ cells, your skin cells, your brain cells, your heart cells, your liver cells, we call those cells diploid if they have the normal DNA content. Recall earlier we said the normal DNA content was 46 molecules of DNA, 22 chromosomes, so 2 copies of each of those, that makes 44. And then the sex chromosomes, X and Y, is 2 plus 44 is 46. Or X and X plus 44 is 44 plus 2 is 46. These germ cells have half the normal amount of DNA, and so they're called haploid. They only have one of the sex chromosomes. In females, they carry an X chromosome. In males, they may carry an X chromosome or a Y chromosome. They have the 22 autosomes, but just one DNA molecule representing each one of those 22 autosomes numbered 1 through 22. And so they have 23 molecules of DNA in germ cells. When I take my 23 and I combine them with my wife's 23 in the stable, loving, monogamous relationship, then that process can lead to fertilization. Meiosis is distinct from mitosis in 2 different ways. The first meiotic division, what we call meiosis 1, is a reduction division. So in mitosis, we have the job of preserving the normal DNA complement. In meiosis, we want to halve the DNA content. We want to divide it by 2. The second thing about meiosis that's very important, and we're going to spend some time on this, is that crossing over is an important feature of meiosis. The non-sister chromatids can swap analogous genetic material. So you have 2 sister chromatids, which are shown here in blue, and 2 sister chromatids from the same chromosome that are shown here in red. At meiosis, there's a feature called crossing over. As the non-sister chromatids touch each other, the same sequence can be broken and reformed, and we make a hybrid sort of chromosome in a process called genetic recombination. So this crossing over ensures that there's shuffling of genetic material, which is an important part of sexual reproduction. Again, making an evolutionary argument, if you think about it, animals undergo a lot of risk to go through the process of sexual reproduction. Sexual reproduction is a really good time to get killed. If you don't believe me, if you're a hunter, when is it that you go out and hunt elk or hunt deer? You hunt elk or deer during the rut when the males are not paying attention to you, and you can sneak up on them and shoot them. We know from this that males are taking some risk when they're going through the process of trying to set up for sexual reproduction. There must be some counterbalancing benefit to sexual reproduction. That benefit is the shuffling of genetic material that occurs during crossing over. during meiosis. Imagine if you got all of your chromosomes that are numbered 1 through 11 from mom and chromosomes 12 through 22 from dad, and just by chance your brother got the same mix, then you and your brother would be identical twins that were born in different years. That would be kind of weird. And so that never happens, and that never happens because of this process of crossing over during meiosis. This isn't just my imagination, although it might be, but we know that crossing over occurs from several lines of evidence that we're going to discuss now. One is that direct photographic evidence, because we can actually see it happening. This puffy thing that we're looking at is a chromosome and a scanning electron micrograph, and we can see the points shown by the white arrows where crossing over is actually occurring along this DNA molecule that's packed tightly in a chromosome.